So we've looked at biological databases. We've seen quite a number of examples in the demo. Took you through UniProt and Protein Data Bank, um, which I think they're quite relevant to this session in terms of molecular docking. So I'm going to talk about chemical databases where you can find a number of chemicals that we use for this technique in terms of I'm referring to the molecular docking. And of course, so many other things that you might want to do when it comes to using molecules and compounds, small molecules. Now, chemical databases are, of course, the backbone of most of our structure-based computer-aided drug discovery, and even ligand-based, of course, computer-aided drug discovery. Um, the central focus of cheminformatics. Okay, and what it does, what's in terms of definition, they are also very organized platform that stores digital information, um, which can be used to build knowledge-based models for drug discovery um, projects. So um, I just want you to take note of the fact that not only are they organized, but you also have a digital format for them. And I don't have enough time to go through the digital format, but I'll just mention a couple of them as I go through this session. All right. So on this page, I put the images from a couple of chemical databases, ranging from PubChem, which is about the largest, arguably the largest um, the chemical database. You have ChemSpider. Um, you also have Chembel. I particularly like Chembel. Is well annotated and yeah, but PubChem is also well annotated. I will show you that. I'm referring to the fact that they have good information for each uh, chemical compound stored within the database. And you also have the drug bank, so which contains all the digital form and information related to uh, all the FDA approved drugs. Okay, so yeah, and particularly dictionary of natural products. Although it's not a free database, but it's one of the most comprehensive for natural products. There are other free database that you can look at. I'll try and put a link for some of the publications that have reviewed natural product databases. And the wonderful thing about this database is they allow you to extract, so just like we're able to get the digital form of the protein and to download it into our computer, local computer, we can do the same with these databases. Allows you to get the three-dimensional structure of these compounds onto your local computer for you to do your simulation as required. So I'm just going to zone in on PubChem because I want to just show you a couple of things on PubChem, and you're going to do your hands-on exercise on this using this particular database. So it's an NIH chemical information resource, and collects data from over 620 resources for each compound. You know, some of them, of course, not some of them might not have all the information, but yeah, for as many that are, they try to get as much information as they can for each of the compound in the database. And of course, it's free. Um, it is claimed to be the largest, and it has a lot of information, especially related to bioactivity that are usually extracted from scientific articles. So basically, if somebody has used that chemical and tested it against a particular disease in a particular bioactive assay, they get data from that with a link to that paper, of course, and incorporate that information into their database. for that particular compound, you know. And other annotation information, sorry, include um, the connection or the connection between that compound and gene and protein targets. So basically saying, um, for the bioactivity that have been tested, what is the potential target for this particular compound, where which proteins does it interact with, and you can imagine if you know the proteins that they interact with, and from that you link that protein to a particular gene, or you link that protein to a particular disease, you can begin to also infer the kind of bioactivity to expect from each compound. You know, so that you can just by looking at databases, there's a lot one can do in terms of research and inferring, um, just not only getting knowledge but inferring some sort of conclusion, as it were, from the question that you ask of that particular database or series of databases. Okay. And it allows you to search. The PubChem platform allows you to search using keywords. If you have the keyword for that particular um, compound, the name, the usual name, the chemical name, uh, anything that's even similar, allows you also to draw part of the structure or the full structure and do a search on that platform. I'm going to show you that. And of course, you can also write just a small script to engage with the back end of the database and to access some compound, especially if you want to access a very large set of compound. You can just write a small script to engage with that. So there is a bar where you can type in, and if you look at, let me just show you this. So you have three tabs there: bioassay, compound, and substance. So under bioassay, you can also write a compound, the name of a compound. It will go under that and search for compounds that have been tested in one bioassay or the other, and of course link that each compound to that bioassay. And of course, if you have just on this tab, it gives you information about that particular compound. Although you will still see a link that if that compound has been tested in any bioassay, you see a link under the information that tells you where all the bioassays that have been uh, tested for that particular compound. All right. Um, so I kind of like this. Although I said I prefer Campbell, I like PubChem because it also has some nice tools that you can use. Um, Bioassay tools, like I said, which is a connection to that each compound. Structure search, um, you can take aspects of the compound that you are querying and say, okay, can you find other compounds that have similar structure to this? And and I want to be able to expand my search space, chemical space, in terms of compounds that are very similar to this. The idea being that if they have the same substructure, they might have the same activity. Okay, so you can do a search. You can convert into a 3D conformation, um, based on the whatever format that is. Uh, okay, I need to speak to you about the format. You can do structural clustering. Basically, this is an unsupervised learning to see, just generate a cluster tree, see which compounds are similar. You know, yeah, yeah, yeah. You can check that using cluster analysis. I don't really want to get into that. Classification model. You can build a classification model. Uh, you can also upload compound. Remember, it's a database. They accept if you come up with a new compound that is not existing, you can upload it, and they will annotate and curate that. Then you can download specific compound. Or use the FTP to download the whole database onto your local drive. That is also possible. All right. You can also, if you look at the search tab, you can enter the name, you can enter what we call a smile string, and you can enter what we call an what I call it INCHI key for each compound. Now, compounds generally have in order to digitize or digitalize compound, the various people have come up with various formats of representing compound. Of course, the structure of a compound that you did in your elementary chemistry. You know what the benzene ring looks like. You can physically draw that on your on a piece of paper. But when you want to translate that to a computer, there must be a way we can represent it such that the computer can read that information and, if necessary, give you and know that it is a benzene ring and give you that information if you require it. So there are various formats um, ranging from smile strings. The smile strings looks something like this. Um, yeah, this is a smile string, and this is a key. There are other formats. Um, there's something called SDF. It can be an SDF format. You can also have more formats. Uh, more, more. So it comes in that file format or mode 2. You have mode and you have mode 2. And you can also store them because if you look at the file you downloaded from PDB, it will have an extension called .pdb. Yeah, it should have an extension. So yeah, and the same way you can store these compounds as with an extension .pdb or .sdf.mol file. If it's a string, this is called a string because it's a series of characters we call smiles. And of course, in this state, it's a specific uh, strings of characters and numbers that forms the key. All right. So you can upload in any of these formats, but you can also upload by drawing the structure. So it has a tool that is attached to it. That's the database, and that allows one to draw the structure. So this is what it looks like. This tool, you can use this to draw the structure of the compound and upload it and do a search. I think you can do that. I'll show you something. So now in this case, the string format was imported. The smiles format was imported. Okay, and gives you if you click on go on that, gives you identity one, similar compound one. Uh, it has one identity for that, but sorry, one identity for that particular compound, but there are quite a number of similar compounds, similar to different degrees. It will tell you the percentage similarity if you explore that. Then those that have similar substructures to your compound, it will also highlight that. Yeah, and some with superstructures. So different tabs, depending on what you want. And of course, you can download a CSV file. A CSV file is like an Excel file, a text file that can be opened in an Excel. Okay, and yeah, and some other information that's related to the gene, the bioassay, and the rest of them for each of the. In this case, there was just one output for this search. And that, I think that's the output. That's the name. Look at some of the information that you also have. Um, Clinical trial information are available, gives information as to which studies have been done using that particular compound, and the status of that study, the status of that such a study. Recruiting, this is the recruiting stage and the phase. Um, this is terminated and the trial was at phase 3. This is completed at phase 1. So it gives you all this information. And it can also give information related to pharmacology and biochemistry, and of course, the occurrence of that compound in literature. And that helps you to see um, how many literature have actually used that compound and for what. Was it for a bioassay or was it used as a reagent? You can also find out. That link is also there. Okay. Okay. You okay? So there is also information about the genes that are related to a particular compound. In this case, actin beta is related to this particular compound that was searched. This is the SMILES format of the compound that was put in the search there. So this particular information is related to the protein to which this compound can bind to, and the gene responsible for that protein. Okay, for the expression of that protein. So such like this. Okay. All right. So we also have information related to chemicals and the bioactivities. Okay. These are the activities: active, active. If it's not active, it will tell you, and you have a link to um, the paper that actually now described that bioassay or that particular activity test that was done. All right. Okay. More information. Sometimes because PubChem is directly linked to the drug bank. So if the compound you search is actually in the drug bank, it gets information from drug bank and displays them, and those information. In this case, uh, it did search for a quercetin and said quercetin, yes. Um, this is the compound CID number, but in terms, it's also a drug. And what class of drug is it? It's an experimental investigational drug. It's not been approved yet. The same thing. Um, these are information regarded to biases. Okay. So what I did here was to list some other chemical databases, quite a number of them. You can just look at them. I try to give very short description of each one of them, and it's good to take a look so you kind of know what is the scope of each database in terms of the compounds that you can find within that data. Some of them they are a very focused set of compounds or a library of compounds. Some are a bit more diverse and very large. Okay. At this point, I think we're going to do a demo for you on PubChem. So let me do that quickly.